I'm going to try to tell you, mostly in the context of genetic association studies, ways in which you can get tripped up using statistics and some things to kind of look out for, and just the basic underpinning of the way that most of those studies are done. Here and there, I'm going to delve a little bit into, dive a tiny bit deeper, but like I said, this is mostly a talk to try to give you a sense of how one should think about the results and interpret results of genetic and genome-wide genetic studies. I'm going to talk mostly about the areas where this has been worked out a little better, which is genetic association studies of variants that are common enough to test one at a time for association with phenotype. But I'm also going to allude a little bit to studies of rarer variation. The detailed methods of those are actually still somewhat in flux, and I'll just very, very briefly allude to those. You may get more of that as the course progresses, and certainly you'll see a lot of that in other MPG talks, different methods for studying aggregates of rare variants, and the statistical issues for those are going to be related to these, but probably slightly different, and I'll allude to that too. Let me just remind myself where I'm going. Yep. Okay, so for this is one of the first phenotypes for which there was success in genome-wide association studies, age-related macular degeneration. And this was very unusual in the field of genome-wide association studies, in that the effect sizes that were observed were quite large. I'm not sure if there's a pointer here. That's right, I'll just, so you can see sort of the, here we go. So the people with the highest risk genotypes at 3 different loci, here compared to the people with the lowest risk genotypes, and this is data from Mark Daly's group and collaborators, have over 100-fold difference in risk. So that's very, very unusual for genome-wide association studies, and sort of the more typical early results of genome-wide association studies are illustrated here, and this is work of Jose Flores, David Altshuler, and groups in the UK and others. So there's a potassium channel, KCNJ11, and there's a common missense variant E23K, and it turns out that for every K or lysine you have instead of glutamate, you have about a 15% increased risk of getting type 2 diabetes, that is a modest effect, but if you type it in enough people, you can get good p-values, and we'll talk a little bit about, actually a fair bit about what good p-values means. So this is the cartoon version of what I just told you. So those of you who know Where's Waldo, age-related macular degeneration would be the goal is to, for those of you who don't know Where's Waldo, the goal is to try to find Waldo in that picture. So this is how easy it is to find complement factor H and associated with age-related macular degeneration. No matter how you kind of do it, it's going to pop out at you. If you're trying to find that E23K variant, though, you have to look harder. And the issue is, once you start looking harder, you can start to find things that aren't really there. Okay, so that's actually, in some ways, kind of the message is in this little cartoon, which is that things that are hard to find, and when you're looking at lots of different possibilities, you can get fooled, okay? And you have to be really careful in order to find what you're really looking for, okay? So, in fact, when association studies started, there was a lot of inconsistency in the field, and this is actually what kind of drew me to this area of sort of horse sense about statistics, and why I'm giving this talk, I think. So we analyzed the association study literature, and we found that at least at the beginning of the field, most associations that were reported by, that were published by one group then didn't replicate when they were attempted to be followed up by a different group. And replication is one of the hallmarks of good science. And so if you're using a methodology where lack of replication is the standard of the field, that's not a good sign. So it was important to figure out what was going on. And it turns out that it was a mix of false positives and false negatives. The false negatives are largely an issue of power, particularly when you apply the statistical thresholds that you need to avoid false positives. But I'm going to talk first about false positives. And they really come from 2 sources. One is just a statistical fluctuation. You think you have a result, but it's actually noise or the result of random chance, and we'll go through that in quite a bit of detail. And then there can also be systematic bias, so that you can get a result that actually looks a lot better than it should. Okay, so those are kind of the two main sources of false positives. I'm actually going to talk about systematic bias first. So I'm just going to, there's really two main sources of systematic bias in a standard genetic association study, and then there's kind of a subtle one that, there are other subtle ones that can come in. But I'm going to talk about the first one I'm going to talk about is due to genetic ancestry. So let me just go through the mechanics of a typical case control association study. This is a little bit oversimplified, but not tremendously oversimplified. In its important respects, this is what's going on. So you've collected cases and controls, and this can be either people with disease and people without, or if you're studying a quantitative trait, you could have actually collected a whole bunch of people who have a range of quantitative phenotypes. And either way, the message here is the same. So what you do is you identify variants, and typically that's a few 100,000 or a million variants or millions of variants on whatever genotyping platform you happen to be using, and in fact maybe even more variants if you're using when you use those to impute genotypes at variants that happen not to be on your genotyping platform. But in any case, you're going to test variants for an association with disease status or with your quantitative trait. And so you might observe something like this, where the T allele is more common in the cases than controls, and this allele frequency, or sometimes genotype frequency, but usually it's done in an additive model, so looking at allele frequency, this difference in cases and controls is the association between the allele and disease status that you're searching for. So actually, before I dive into ancestry, I'm just going to point out that genotyping quality is also a very important source of artifact. And when you're doing QC of your association studies or of any of your genetic studies, QC of your genotype calls is really important. So this is one way, and this is pointed out by David Clayton and colleagues, one way in which you can get a false positive association. So imagine that your cases and controls, for whatever reason, are of slightly different quality of DNA. And this is actually sometimes can happen where the cases have been collected over 20 years, and then people decide to do the study, and then they go collect their controls. And the controls might have been collected in a different way. And imagine that the heterozygotes, and this is often the case, are actually a little bit more difficult to genotype. So you get a no call a little bit more often than you do for the other genotypes. And there's often an interaction between DNA quality and genotype calling, so that the DNAs of lower quality will be slightly more likely to have a no call than the DNAs of higher quality. So what you'll see is you'll see a dropout of some of the heterozygotes, and that'll be more in the cases than the controls. And if you do the counts and everything of what I've illustrated here, you'll see that actually now the T has a higher allele frequency in the cases than in the controls. So it looks just like the other example of the association you're looking for, but this is entirely an artifact. And the way you can tell this is an artifact is, first of all, you can apply very stringent thresholds for calling the genotypes. And the other thing you can do is that oftentimes these dropouts will differ from laboratory batch to laboratory batch. So one of the other really things that turn out to be really important in QC of association studies is to look actually for an association with genotyping failure and plate. And then the last thing is that this genotyping failure will usually be specific to that particular SNP and not necessarily to the SNPs nearby. And so if you have SNPs nearby that are in linkage disequilibrium or correlated with the SNP that's having the selective failure, you can predict when that genotype is going to fail based on the genotypes of the surrounding variants. And that's, if you happen to use Plink, it's something called mishap. But basically, that's another sign that there's a problem with that particular SNP, and maybe you shouldn't actually pay so much attention to that SNP. Because if you ignore all of these QCs, then what basically ends up on the top of your list of the most associated SNPs are all the various different ways in which genotyping or the other artifacts I'm going to tell you about can confound your results. And you're basically enriching very, very strongly for those bad behaving SNPs. And so if you don't pay attention to QC, the top of your list is going to be exactly the things you don't want to look at. Okay, so that's genotyping artifacts, and they're other things that can come about when you do imputation, but I'm not going to really go into those. So this is now talking about confounding by ancestry, which is kind of the other main way in which association studies can get confounded. So imagine if you, here again, you have your cases and controls, but now instead of ascertaining them from a single homogeneous population, you're actually ascertaining them from multiple populations. Now, if you've done this and everybody's self-identified correctly, saying I'm population 1 or I'm population 2, then you can actually just do 2 different association studies, get the summary statistics, and then combine them using meta-analysis, and there are all sorts of programs like Metal and many others that allow you to combine the association statistics in a statistically valid way that really loses almost no power when you do that combined analysis. The difficulty is that self-identification is not always correct, and so what you often end up with is a mix in your cases and a mix in your controls of multiple different populations or people whose genetic ancestry vary slightly. But the labels are not sufficient to distinguish them. So if your diseases are of equal prevalence across all populations, then you don't have to worry, and similarly, if your quantitative trait distribution is the same across all populations, you actually don't have to worry about this, but that you often don't know whether that's in fact true. So imagine that you have 2 populations, green and magenta, and the green population has a high prevalence of disease. So your cases will be enriched for people with green ancestry, and your controls will be enriched for people with purple ancestry. If you now type a genetic marker that happens to differ in allele frequency across populations, and pretty much for most pairs of populations that are even separated by a tiny bit, like say Northern and Southern Europe, you will be able to find markers that are different in frequency between 2 populations. And so for these markers, what you'll see is that the orange allele, which is common in the green ancestry, will look like it's associated with disease, but really what's happening is that this orange allele is just a marker that's telling you that you didn't match your cases and controls properly by ancestry, and that there's more green ancestry in the cases than there is in the controls, and the orange allele is just a readout of that ancestry. And so this is a false positive association, is due to stratification. And as I'll show you, this has been reasonably well dealt with, although maybe not perfectly, in the common variant association studies, and it's something that's just probably going to rear its head again as we get to sequencing studies, which have even more differentiation between populations when you go down to the very low allele frequency range. And if you sort of think about it, very common alleles are very old, and they were sort of seeded into most populations around the world, or at least most reasonably closely related populations. They're probably seeded at pretty equal frequencies. New, very rare variants might be so new that they actually post-date a split of what would otherwise be pretty closely related populations, and because of genetic drift, they might have vanished just by chance from one population and been maintained in another population, and so the differences in ancestry can actually get more significant when you're dealing with rare variants, and I'll try to come back to that a tiny bit.
Joel,
yeah.
For a second. Aren't those increasingly baked into the quality control of these things? In other words, early on this was a big problem, but over time, don't you have certain markers that alert you to that, such that you send off a yellow flashing light to say this is
So if you know if you know ahead of time which ancestries are in there, then you can, and you know which markers differentiate those ancestries, you can look at those. Although it actually gets complicated because if you have a phenotype that truly is genetically associated with a cline, then you expect those to be enriched in your association. I'll describe what's been developed and sort of how well it does. Okay, so just to give the classic example of this, so height is one of the things we study. It's highly heritable within populations, but height actually varies widely across Europe. And in fact, we actually now know that this is in fact due in part due to genetic differences at variants that have different allele frequencies in Europe, probably due to recent selection. You can just out in Nature Genetics for those who are interested. But we actually used height as a way of demonstrating stratification within what was at the time thought to be a homogeneous population, which was Americans of European ancestry. So height, as I told you, varies with European ancestry. People who are tall tend to have more Northern European ancestry. People who are short tend to have more Southern European ancestry, and that's shown there, where we had designed a panel of tall and short European Americans. And there's a variant at the lactase locus, for reasons that I don't have time to go into, also varies a lot in frequency across Northern and Southern Europe. And again, given any two populations, you can usually find variants like this. So if you type lactase and the persistence allele, which is the allele that lets you digest milk into adulthood, in this group of tall and short European Americans, what you'll see is that the Northern European ancestry folks will be tall and have a high frequency of lactase persistence, and the Southern European folks will have the opposite, and so lactase persistence will appear to be associated with height. And we actually kind of knew what the answer was going to be before we even did the genotyping experiment. And the reason we knew the answer was because this is actually a false positive association. We'd set up confounding by ancestry, but in fact, this happens, you don't have to set this up. This happens all by itself if you just kind of take a even modestly mixed population and look at any phenotype that happens to be a little bit variable along that same axis where you have ancestry mixture. Okay, so how does one diagnose this sort of problem, and how do you correct it? So Bill referred to some flags that might come up. So there's one flag, which is a useful initial flag as a diagnostic tool, called genomic control. And so what this is typically called lambda, and this is from Devlin and Rader, and then David Reich's group with David Goldstein as well. Essentially what you do is you take your p-values and you convert them to chi-square statistics, and that's something that's pretty standard to do. You can even do it in Excel and R and a whole bunch of other things. And you take the median value, so the best p-value, the worst p-value, and the one that's right in the middle, that's your median, and you compare that to what the null distribution would be, so just a chi-square distribution sampled at random. And so under the null, the median chi-square statistic with 1 degree of freedom should be about 0.48, and you can then say what's the value for my actual association study, and then you take that ratio, and for a nicely controlled association study, if you have about 1,000 people, 1,000 cases and 1,000 controls, really it should be well under, say, 1.1, and really 1.0 something low is kind of a usual performance. This, however, does, even in the absence of, and if you have a value of, say, like 1.2, that tells you that there's some systematic inflation of your test statistics. Okay, and something is probably wrong with your association study. It might be confounding by ancestry. It might be technical artifacts. It might be that there's like an extra comma in your file and everything has gotten thrown off. Could be lots of things. But this is sort of a useful diagnostic tool. And this can, in theory, broadly adjust for stratification. A lot of people use it that way. They say, well, we detected some stratification because the lambda was 1.1, but then we divided by 1.1, and then that we divided all our chi-square test statistics by 1.1, and now the median is 1, which it has to be because that's what you've just done. And so now there's no more problem with stratifications. That's not true, right? All that's done is sort of deflated your test statistics by the amount that they were inflated, but your rank hasn't changed. And remember, what I told you is that if you have problems in your association study, the things that are at the top are enriched for the problems. So the problems are still at the top, they're just not quite as significant and being at the top. It can actually deal pretty well with cryptic relatedness due to cousins and stuff like that, because that's actually kind of a random genome-wide thing, and that's it's not locus. It's not, assuming you have lots of families, it's actually not concentrated at one locus. And I did say this scales with sample size. And one issue that's come up, and particularly actually in height, it's kind of the most glaring, is that as you get larger and larger association studies, so not just 1,000, but many thousands or even hundreds of thousands, lambda scales with sample size. So you can actually get, just due to real signals, you can get lambdas of like 1.5 or 2 even, which would be something that, when we started this, we would have been horrified and just basically thrown all the data away and not looked at it again. But in fact, you can show, and Peter Vischer's group has done this, that in fact you expect this sort of inflation if there's substantial polygenic inheritance, because there's so many loci that are actually associated, some of them very strongly, and then due to correlations, due to LD, you get a lot of other SNPs nearby that also show some association. That's enough to throw the entire distribution off. So your null distribution isn't really a null anymore. The genome-wide distribution is no longer null, and that makes it even more difficult to use lambda to interpret your results. So fortunately, there are better tools to deal with stratification, and you can use lambda a little bit to assess how well they're doing. But again, because you're not necessarily, as your study gets bigger, it's not clear exactly how close to 1 you should be getting. So it's a little difficult to, again, figure out whether you've done a perfect job. But anyway, this is an example of principal component method, and a common implementation of this is something called Eigenstrat or Smart PCA is part of that, or there's similar programs in Plink, which is like the MDS multidimensional scaling, is also essentially principal component based method. And what this does is this takes dense genotype data, so lots of SNPs all around the genome, and basically the idea is that if there's variable ancestry, there'll be subtle but correlated shifts in allele frequency in the people with one ancestry as opposed to the people with the other. So like, for example, Northern and Southern Europe, lactase is not the only variant that differs between Northern and Southern Europe. There are lots of variants that differ, and what you'll see is that there'll be kind of a correlated pattern, so that in one Northern European, the alleles that they tend to have will be enriched for the Northern alleles, and in another Northern European, they'll be enriched maybe not for the same exact alleles, but some of the alleles will overlap, and so the enrichment or the differences in allele frequency will be correlated in the people who have similar ancestry. And the one way of doing that is to use principal components, and I'll show you what that means in the next slide. But basically, this is a plot of European Americans, I think from the Framingham Heart Study, if I remember right. And so what you see here is 2 different principal components. So essentially, this takes those correlations and breaks them into independent groups of essentially axes of ancestry. And so here's the first axis of ancestry, and this actually corresponds to the Northern to Southern European axis. So there's mostly people of Northern European ancestry, but then it goes down into people of Southern European ancestry. And actually this cluster up here is one that's common depending on where you do your study in the United States. That's actually probably mostly Ashkenazi Jews who are in the upper left corner. And so, if you didn't correct for this, what these axes are telling you essentially is that they're correlated allele frequency differences among this group versus this group, and this whole group versus this group. And if there are any phenotypic differences between the 2 groups for whatever reason, then those will be correlated with those same axes of ancestry. But because these are all correlated, what you can then do is you can actually use these principal components essentially as covariates in your analysis. And that's what the advance of Eigenstrat was to realize that. And if you use those as covariates, and if these have adequately captured the ancestry variation in your study, then this will remove the problem of stratification, or at least largely remove it. So this is, again, just principal components, and this is pretty much what I said, and there are many similar related methods. But essentially, you take the variance in a data set and break it down into orthogonal, which really means statistically independent components, and these are some of the ways it's implemented. I should point out that there are actually newer methods now to try to control for stratification, and there's some debate as to whether principal components actually really do a perfect job of removing ancestry, confounding by ancestry or not. And our experience has actually been that it actually does pretty well still when we try to set up all sorts of ugly stratified scenarios. But there are other methods which involve stuff called mixed linear models, which I'm not going to really go into. But just to point out, one of the first tractable or computationally feasible methods is called EMMA-X, and there are actually many more recent, faster methods. So if you just kind of look up mixed linear models and ancestry in PubMed, you'll get a whole slew of these. OK, so there's a lot of experience now with this with common variants, and like I say, using things like principal components and possibly these mixed linear models is a way of avoiding stratification. And actually, one thing I forgot to say is that if you happen to have a family-based study, though you can do tests that are completely immune to stratification, although you have to essentially throw out the phenotypes of the founders in order for it to be completely immune to stratification, so it's a little bit less well-powered, but it can be another useful check if you happen to have that. The other thing to watch out for is that family-based studies are particularly susceptible to genotyping artifacts, and again, sort of a whole other topic. Okay, so rarer variants are really going to fall into two classes of analysis. One is going to be one variant at a time, and one is going to be grouped variants. And so one variant at a time is basically what I've just been telling you about, with those little circles and looking for the orange allele being more common. And the only difference here is that the variants, instead of being 5 or 10 or 20 or 30% allele frequency, they might be 1% or 0.5% allele frequency. But if you have enough individuals, you can actually study those variants one at a time. So the challenges are, so some of the challenges are technical. So because these are rare in any given usual size study, the number of actual minor alleles that are present in that study is low. And as I think I'm going to get to show you at the end, if you have low minor allele counts, sometimes that can give you very unstable statistics. So you have to be wary of the method that you're using here, and you might want to look at sort of what's the distribution of p-values of all of the variants that have that kind of minor allele count, just to make sure that the statistical package you're using and the way that you're implementing it is actually giving you the kind of expected results. So if you do this and you look in the typical p-value for something with low minor allele count is 0.03 as opposed to 0.5, then that tells you probably something is not right with your analysis. Again, there aren't going to be that many positives in the genome, and this is sort of a general principle that if you start to look at a particular class of variants, whether that's variants across the genome or variants of a particular allele frequency, looking at the distribution of results for that entire class of variants is a very good sanity check. The other thing is that it's harder to genotype rare variants, although genotyping keeps getting better and better. But this is sort of one extra little bit degree of difficulty. So again, you want to be more worried about genotyping artifacts, doing things like going back to the actual genotyping data and looking at the clusters to see if there's something weird going on. Looking again for those plate-specific artifacts, all of those, making sure that the call rate is high. If there are correlated variants nearby, looking at those to see if they at least show some signal, and maybe even re-genotyping your best results to make sure that they're actually correct. Then there are going to be some variants that are so rare, they might only be seen once, or maybe once in a couple of different populations. And so you're never going to get any statistical evidence for that one variant by itself. So you're going to have to study those variants in aggregate. So the question here then becomes not, is this variant associated with a phenotype, but is a group of variants of a particular type within a gene, say, or a pathway, is that associated with a phenotype? And so there are many methods now, again, that are, and there's sort of just a couple listed here, and I apologize for not having put the references in. But the SCAT, you can just PubMed variable threshold. This actually comes from Alkes Price and others, and there are many other methods. And a lot of these methods are looking for a signal where the rare variant, so the minor allele, is associated with a change of phenotype in one direction. So it might be associated with case status, or it might be associated with high phenotype value or something like that. And then there are also, and this is from Mark Daly's group and others, it's called C-alpha. So basically where you're looking not for a shift of the minor alleles, but an excess of the rare alleles in both tails of your phenotypic distribution. So for example, both cases and say hypernormal controls, or both tails of a quantitative trait, have an enrichment of minor alleles, and there's a way you can look for that to say, usually you'd expect your minor alleles to be distributed evenly across your distribution, and if they're sort of clustered to the tails, you can do a statistical test that will give you a signal in that scenario, and you can imagine this sort of a loss of function and gain of function alleles in the same gene. And there's still a lot of work going on to figure out which of these tests is best powered and least susceptible to various different artifacts, and under which different, and probably the answer depends on what's going on in an underlying genetic model and what your mix of ancestry is and all that sort of thing. And then finally, one way that may end up being very useful to group variance is if there's functional evidence, and this could be either bioinformatics, so looking at for loss of function as opposed to missense, or it could be direct functional evidence and just taking those variants and grouping those. And one thing I'll say about this is that because there's in theory, lots of different ways you could do the functional work, it's usually a good idea to have laid out what the functional criteria will be ahead of time before you go grouping your variants, because if you get your results and then decide, okay, these are the variants that I see in the upper tail, oh look, they all have leucines in them or something like that, so I'm now going to group my variants by the ones that change the amino acid to leucine, right? That's not a fair way of doing the test. You have to have kind of decided how you're going to group your variants ahead of time before you look at your results. And if you don't do that, then you have to really, then the onus is on you to replicate it, taking that one specific hypothesis and testing it in an independent population. There also may be problems with rare variants in ancestry. As I talked about, rare variants are more likely to be private to particular populations. And when you're looking in aggregate, then actually a different sort of ancestry confounding comes into play, which is if you have populations which have differences in overall nucleotide diversity. So for example, African ancestry populations have higher nucleotide diversity, and founder populations can have different patterns of nucleotide diversity as well. And so if your cases and controls are mixed for that, and we actually, in our lab, again, unintentionally, had a very, very nice demonstration of this where we had some really lovely results that were completely due to not having quite perfectly matched our cases and controls, even though we actually did it to some degree. So you have to watch out for that.
What do you mean by nucleotide diversity?
So, for example, African ancestry populations, because they're older populations and haven't gone through bottlenecks, they actually have more rare variation than non-African ancestry populations. So if you just take a gene, and let's say your cases are all African ancestry and your controls are all European ancestry, every gene in the genome will look like it has an excess of rare variants in cases versus controls.
And it's genomic diversity, yeah? It's not
Yeah.
too many G's or C's or T's.
No. It's variation. Yeah. So diversity at each position is more likely to be diverse. Okay, so association studies really have a couple of key features. I pointed out kind of the modest effect sizes, and I haven't had time to give you my spiel about why we care about modest effect sizes. I'm assuming that's taken for granted here. But you need power to detect these. But as I illustrated with that cartoon, once you start having to look really hard for things with modest effect, there are all sorts of other things which can have modest deviations in allele frequency. They're not due to true association, and so you have to be careful about your quality control. And so, again, if you don't have enough power, you can get statistical fluctuation for false negatives. And the question then becomes, what should be the power threshold? What should be the statistical threshold that you need to use to declare an association as positive? So I've been talking about systematic biases. But now, if you've done your study perfectly, where should you declare victory and where should you not? So to illustrate this, I'm going to switch to a baseball metaphor. And as sad as it is this season to have to put that logo up here, I still will use the Boston Red Sox. So I apologize to those who are not baseball fans for this next portion of the talk. But imagine that Bobby Valentine has been fired. Not too hard to imagine. And you have been picked as the replacement manager because nobody else wants the job. And your first job for 2013 is to pick the designated hitter for the Boston Red Sox. And you have spring training to decide. And the way you've decided you're going to do this is you want a lot of home runs. So designated hitter, for those who don't follow baseball, his only job is to hit. Okay? And so you decide that the person with the most home runs is going to win, and each candidate is going to get a certain number of tries to hit home runs, a certain number of at-bats. So this is the current designated hitter. Despite his current Achilles injury, this is probably who you want to pick even for 2013, assuming they re-sign him. But you don't know what David Ortiz looks like because you're new to baseball, and so you could get fooled. So you can't just pick him. You want to do sort of a fair trial, so you don't get some guy doesn't come in wearing an Ortiz jersey and you say this is the guy. And you certainly don't want to pick this guy, right? Your whole goal is to pick somebody who's better than the average. Okay, so this is kind of like your null distribution. I've used poor Mike Aviles as the demonstration this year. So Ortiz has been the same every year I've done this, and this person has been traded every year. So this year it's Mike Aviles, not making any predictions about whether he'll be with the team next year. But in any case, if David Ortiz over the last few years has had a rate of 0.06 home runs per at-bat, and Aviles has been at the major league average, which is 0.02 home runs per at-bat. So if you gave them each 50 at-bats, which is actually nice long spring training, you'd expect the typical result to be about 3 home runs for Ortiz and 1 for Aviles. So you can say, well, is that actually a significant difference? And so essentially what this is a 2x2 table, a 2x2 called contingency table, and which you can, you can calculate a couple of things. One is you can calculate something called an odds ratio, which is kind of the ratio of the odds of, sort of how much more, it's kind of usually used as a measure of effect size. And in this case would be about 3, not quite exactly 3. And then you can compare the observed table to an expected table and calculate a chi-square statistic, and that's shown here. And for those of you who are jumping up and down and saying those numbers are too small to do a chi-square properly on, you're correct. You would actually use a Fisher's exact test for that table, but the principle of comparing the two tables is the same. And what you can then get is you can get from this chi-square statistic an estimate of statistical significance to see whether that 2x2 table is different than what you would expect under the null assumption that Ortiz is the same as Aviles, so that Ortiz is like the null, Ortiz is like an average Major League Baseball player. So if you do this, you would actually get that with 50 at-bats, even with the typical expected outcome, you would not get a statistically significant difference. So 3 home runs is actually not statistically different from 1. This is, even if Ortiz were a kind of average player, him hitting 3 home runs would actually not be that unexpected. Okay, so how many bats, oh, I'm sorry. So just a slight divergence, you can actually say, well, maybe just home runs are too rare an occurrence to use as my outcome. So you could do some quantitative traits, like how, if even though he didn't hit a home run, he hit it right to the fence, whereas Aviles kept hitting little ground balls to the infield. And you could do all sorts of quantitative traits, and this is kind of what association studies do also. They study both disease outcomes and quantitative traits, hoping that the quantitative traits are actually in some ways a surrogate for the disease outcome that's a little rarer. And you can do this using regression, and in fact, for both the quantitative trait and the dichotomous, or the yes-no trait, the regression-based framework is really the way that most people do this. And essentially what this is you model your phenotype y as some mean value plus a set of betas, which are the regression coefficients, times x sub I, which is your genotype, essentially, or when you're doing this in the genetic context. And your null hypothesis is that beta is 0, which would mean that it doesn't matter what your genotype is at this locus. Okay? And regression can take into account all sorts of covariates. So for example, if you had always played Ortiz in the day games and always played Aviles in the night games, then you might see a difference, but that's because it's easier to hit home runs, actually I don't know which it is, probably at night, than it is during the day. And so you would take that into account, and there are ways of doing that in the regression framework. And this is linear regression for quantitative traits and logistic regression for dichotomous traits. Okay, so that's kind of how you would do it. You wouldn't necessarily really just do these 2x2 tables, but it's a good approximation. So how many at-bats would you need to see a statistically significant difference? So if you had 200 at-bats each, which is actually a good chunk of a season, you can sort of get a barely nominally significant result if both have the typical outcome. And I should say this is not actually how you calculate power. You don't look at the typical outcome. There are power calculators that you can use to do this, and some of those are listed here. All right, and I'm sorry, this is basically what I just said about quantitative traits, so I'm just going to skip over this. So this is kind of one of the key points that I want you to take home. So imagine that you're in the situation where it's not just Ortiz versus Aviles, but people hear that there's this sucker who's the Red Sox manager, and there's this opportunity to get in to be the DH of the Red Sox. And so you get 1,000 people who are the equivalent of Mike Aviles to try out. So they'll average 0.02 home runs per at-bat, but if you give them 200 at-bats, some of them will get lucky and hit a lot more home runs. Some of them will be unlucky and they'll hit no home runs at all. And they'll fluctuate up to and maybe even over your Ortiz level, which is kind of what you're looking for. which is 0.06 home runs per at-bat. And then furthermore, your true positive here, Ortiz, might not hit exactly 0.06 home runs per at-bat. So if you just took the best person and you had 1,000 people trying out, the best person probably wouldn't actually be David Ortiz. It would be some Mike Aviles who happened to get lucky in those 200 at-bats. And if you gave him another 200 at-bats, he'd revert right back to form. So this is kind of like the importance of replication also. So then consider the awful scenario where actually, unbeknownst to you, the team did not re-sign David Ortiz, and all you had was these 1,000 Mike Avileses. So the same thing's going to happen, right? So you'll have this, and some of them will appear to be David Ortiz-like, and you will pick them. Okay, so this is basically saying that even though 200 at-bats is enough to tell the difference between one really good person, or one true positive, and one false, one sort of null, if you have 1,000 nulls, then the number of at-bats you need actually goes way up. So essentially, you need to have more than just that modest statistical significant difference of 0.04 between the two to account for the fact that 1,000 people have tried out. So this is the multiple hypothesis testing problem. Essentially, each Mike Aviles that you try out is a different hypothesis. You're saying the hypothesis is this person is different than the null. This person is different than the average Major League Baseball player. And so how do you account for the fact that you've done that test 1,000 times, and one of those 1,000 times you're going to get a p-value of 0.001. That's kind of what a p-value means, the likelihood of observing that result by chance. So this is basically what I just said. This is the hypothesis. You compare the results, but then you need to use stringent statistical criteria. So how do you correct the p-value for the number of batters that you've seen? And one very common way of doing this is called the Bonferroni correction. This is assuming that each statistical test is independent, which in this case it is. Each batter doesn't influence how the other one does, presumably. And so it turns out that you basically multiply your p-value by the number of tests you've done, and that's because of this statistical approximation here. And so that's where the Bonferroni correction comes from. So bringing this back to genome-wide association studies, it turns out that for European ancestry populations and common genetic variants, there are about 1 million independent tests. And what's independent is also a little bit tricky to figure out because there's partial correlation between lots of SNPs. But if you estimate that, they're about 1 million. And so if you work out that you want a p-value of 0.05 after correction, the critical threshold is 5 times 10 to the minus 8th. You can also assess your significance by something called permutation, and sometimes this is necessary when you can't quite disentangle that correlation structure and figure out how many independent tests there are. And so what you do is you permute your cases and controls and redo your analysis hundreds of times. You take your best result each time and you compare your actual best result to the permuted set of best results, and then you can get an empirical permuted p-value. And things like Plink often have ways of doing that. Obviously, this is computationally a little more intensive, and you need the individual level data to do this. So I just want to talk briefly about Bayesian approaches, and then I'll finish up. So there's a different way of thinking about how do you interpret your results. So what I showed you now is a frequentist interpretation, which is you say, how many tests did I do? Let's correct for that, and then that's my significance. There's a problem with that, which is that if you then do one more test, in some sense you have to then go reinterpret. So let's say you only type 10 SNPs, and I say, well, I'm only going to correct for the 10 tests that I did. In some ways, that's a valid experiment-wide significance, and it gives you and the reader of your paper a chance to figure out what was the likelihood that your experiment would come up with that result. But it doesn't necessarily give you the real likelihood that that's a true result, because you could, in theory, have tested the whole genome. So Bayesian approach is a different way of trying to do that. And essentially what this is you start with a prior probability that your hypothesis was correct. You then re-inform that probability with data, and you get a posterior probability. So here it is for association studies. What you're looking for is the probability that your variant is actually causal or contributes to your phenotype, given that you see an association with a certain p-value. And so if you apply Bayes' theorem, it works out to this. And basically, there's a bunch of different terms in here. This is your prior probability that the variant was causal. This is the probability of seeing an association that was causal. That's basically power. And then this is the probability of seeing an association at a certain p-value if it was not causal, which is basically the p-value. So that's your probability of seeing something by chance. And I'm sorry, I forgot this was animated. So the tricky part of that, so power is a little tricky to estimate because you have to kind of guess what the effect size is. P-value is straightforward. What are the prior probabilities? That's what's kind of tricky. And so, this is actually probably how managers really pick their designated hitters, right? Because they don't have 200 at-bats to try them out. And they start with a prior probability of knowing the guy and saying, this is a high prior probability. Mike Aviles is a low prior probability. And then, this you really want to assign the prior to 0 to make sure that no matter how many home runs I might happen to hit in spring training, I'm not picked. Okay, so in action, this is work that Chris and I did now a number of years ago, just taking a bunch of different models and saying, well, what if you had sort of a random variant or a candidate, good variant in a candidate gene or something that made just incredible sense, and assign them different prior probabilities based on some genetic models. And this was the optimistic scenario. And so even under the most optimistic scenario we came up with, a p-value of 0.05 in an association study, even if you just tested that one variant, was almost certainly incorrect. And if it was not a great variant, it was definitely incorrect. So p-values of 0.05 in this setting, and in any setting where you're doing this kind of multiple testing, or where the prior likelihood of any one hypothesis being correct is low, 0.05 is just not a good threshold. Okay, and then under sort of more realistic models, what we ended up with actually was right back at our 5 times 10 to the minus 8 threshold for a random variant from the genome. So both the Bayesian approach and you can do sort of more formal Bayesian approaches than this. But the Bayesian approach and the frequency approach actually sort of coincide on the same threshold. So 5 times 10 to the minus 8 has become really the standard threshold. And it's actually not clear what the threshold is yet for rare variants, and it probably depends on how you're doing your test and what group of rare variants and how you're, in functional classes and all that sort of stuff. I'm going to skip through, I think, some of this. Yeah, so this is just the one point that I made. You can use, so in terms of accounting for relatedness, there are methods that are used for family-based studies. Some of these are unstable at low allele frequencies, so just watch for that. And it's best to compare to methods where you've just ignored relatedness as a sanity check. For those who do meta-analysis, there are all sorts of issues related to strand flipping. This is not a different artifact than genotyping problems, so just watch out for that. And then I talked about some of the problems with rare variants where you can have unstable statistics, especially with dichotomous traits or family-based regression method. Imputation can be somewhat harder to do, certainly for rarer variants, so I don't think we have a tremendous amount of experience with this yet. And then you want to make sure that your phenotype is actually relatively normally distributed, so regression models assume normality of your phenotype. If it's non-normal with some substantial outliers, and I'll show you one example of this at the very end, then you can get really fooled. I talked about some of these challenges from the sequencing era. Yeah, so this is the example. So this is a real-life example. We were the analytic pipeline for a project called CARE, and we would get all sorts of combinations of genotypes and phenotypes to analyze. And this was one of the QQ plots that we generated from a family-based approach. And it was clear that this was either like the best phenotype ever to study, or something was wrong. So this is the expected, this is a quantile plot, this is the line of what you would expect under the null, and these are the results ranked in order of getting better and better by p-value. And you can see there's a huge excess of things over the null. And then that turned out to be due to 4 phenotypic outliers, and when we removed those 4 phenotypic outliers, now everything made a lot of sense. So it's not just at the genotype level that you have to be careful about your QC, it's also at the phenotype level. And again, particularly small numbers of minor allele counts and or family-based methods are susceptible to these sorts of problems. And there are ways of transforming your phenotype, but one of the safest is actually something called the inverse normal transformation, which is a rank-based non-parametric transformation where you basically force your distribution to be normal. So in addition to power and quality, which I talked about, the other 2 things are rigor and skepticism. Basically, if you get something, a result that seems too good to be true, you should be, your first thought should be, where is the artifact? What is the problem here? If you keep that in mind, you're less likely to go astray. So thanks for your attention.